Greetings, biologists. We are Thursday Lab Group A.
My name is Emily Adams, and I'm a biomedical sciences major.
My name is Emma Boyles, and I'm a biochemistry major with a minor in biology.
My name is Peyton Farnsley, and I am a freshman microbiology major with a minor in chemistry.
My name is Dolan Freeman, and I'm a freshman biological sciences major with a minor in chemistry.
Stomata are defined as many of the minute pores in the epidermis of a leaf of a plant, forming a slit of variable width which allows movement of gases in and out of the intercellular spaces. In other words, stomata are small pores that have many uses. Stomata are located on the underside of a leaf. Stomata are enclosed by guard cells, which regulate the opening and closing of the pores. One of the roles of stomata is controlling the rate of transpiration. The rate of transpiration is defined as the amount of water lost by plants over a given time period. The transpiration of a plant increases when the stomata are open and decreases when the stomata are closed. The rate of transpiration works to regulate the internal temperature through evaporative cooling to keep the plant healthy. Water uptake, which is another role of the rate of transpiration, works to absorb nutrients from the soil. Stomata also control gas exchange between a plant and the atmosphere. There are two primary gases that enter and exit the plant through the stomata. Carbon dioxide enters the stomata and oxygen gas exits the stomata, and both of these gases are important for photosynthesis. This process is important because water and nutrients that the plant need can be lost if the stomata remain open for an extended period of time. Stomata are also important in ways that they are indicators of air quality and levels of pollution. The more stomata there are on a leaf, the lower the carbon dioxide levels in the atmosphere. Carbon dioxide is one of the gases that contributes directly to climate change because it is a greenhouse gas that gets trapped in the atmosphere. So if a plant has a high amount of stomata, the less carbon dioxide there is in the immediate area. This is also why there are many initiatives to plant more plants and trees, because the control of greenhouse gases is necessary to control climate change.
Our plant for the experiment was an American holly, which was located on the south side of the biology building. This plant is commonly referred to by its common name, American holly. Its scientific name is Lex opaca. It is of the eukaryote domain and from the kingdom Plantae. Its phylum is Spermatophyta. American holly is native to the eastern and south central United States. American hollies will typically grow to a height around 15 to 30 feet tall. A mature holly can even grow to be around 100 feet tall. For our experiment, which is comparing the stomata densities in two situations, we decided to use a holly bush, which has one side in the sun and the other covered in shade. We decided that our hypothesis for this experiment would be that leaves in the direct sunlight would have lower stomata densities than the leaves in the low light. In simpler words, the more sunlight there is, the less stomata. To get the stomata counts we needed, we first had to make stomata impressions. This was done by painting a small amount of nail polish onto a leaf and then pulling it off with tape and placing the impression on a glass slide, and then viewing it under a microscope to count each stomata. The highest number of 3 stomata counts per slide was recorded, and the average of the stomata counts from 12 leaves in the sun was compared to the average of the 12 leaves in the shade.
Our prediction for the experiment is that a direct sunlight lowers stomatal density, then the holly leaves facing the sun will have less stomata, meaning that the leaves taken from the sun will have a lower average number of stomata per millimeter squared, and the leaves in the shade will have a higher average number of stomata per millimeter squared. the stomata per field of view were counted and produced the results displayed in the graph shown. The stomata counted per millimeter squared were then averaged, and these results compared to our prediction. Our results from the calculated average supported our prediction. The average stomatal density of the plants in the sun was 77.95 millimeters squared, with a standard deviation of 18.46, while the average stomatal density of the plants in the shade was higher at a value of 83.3 millimeters squared, with a standard deviation of 11.70. The averages are displayed in the graph, showing that the leaves in the shade has a value that, while slight, is still higher. We used Excel to run a t-test on the average values for both the leaves in the shade and in the sun. The t-test produced a p-value of 0.40, which is greater than a p-value of 0.05. The p-value shows that 40% of the time, the difference between the leaves in the shade and the leaves in the sun was as big or bigger than the difference that we observed. This shows that there is not a statistical difference between the values obtained for the leaves in the shade and the leaves in the sun.
With an increased effect of climate change and growing levels of greenhouse gases, such as carbon dioxide in our world, stomata densities can be used to measure and compare carbon dioxide levels in different locations. In conclusion, our experiment displayed how our hypothesis of higher amounts of sunlight causes a lower density of stomata in our American holly plant was true. The area of the holly that were shaded had a much higher stomata density than the leaves in the front who receive sunlight throughout the day. This displays how location to the sun can affect stomata density on plants. We would like to conduct a future experiment testing how location can cause plants of the same species to grow at different rates due to stomata density. This experiment could be conducted here on the Southwestern Oklahoma State University's campus with a variety of species. Experiments similar to this one have been conducted before, but to our knowledge, one has not been done here on the SWOSU campus. We have also raised certain questions, such as, how does a lower stomata density affect the O2 production? And how is photosynthesis affected by lower stomata densities? We hope to continue research and have the ability to answer those questions based upon scientific data. Our sources are displayed on the Works Cited page. We dissected these sources and withdrew information to increase our understanding of the mechanisms involved in the stomata processes. Thanks for watching.